Hello all, welcome back to Chapter 3. So in the previous lectures we talked about what is a neuron and how signals are sent through a neuron. So that would be a nerve impulse. Now in this lecture we're going to talk about how cells talk to each other in the nervous system. So the first thing you're going to want to know is what a synapse is. So a synapse is the connection between two neurons. So this is where the axon terminals of one cell meet the dendrites of another cell. An important thing to note is that neurons do not physically touch each other. There's actually a very small gap in between the terminals and dendrites, and this is what we call a synaptic cleft, I'll sometimes refer to it as the synaptic gap. But the technical term is the synaptic cleft. And so when we're talking about a synapse, what we're talking about are functional connections between neurons and their target. Their target is usually another neuron. However, it can be a glial cell. So remember, those are those helper cells. It can also be a functional connection between a neuron and a muscle. And that's how we get muscles to move and contract, is through these synapses. So this is what a synapse can look like. If you notice in the top figure here, then you have what's called the presynaptic neuron. So that's the first cell that's sending the signal. And then we have a postsynaptic neuron. And so the postsynaptic neuron is the neuron that's getting the signal. And you can kind of see here how these synapses are very tiny, and when you zoom out, you can't see that gap in between the axon terminals and the dendrites. However, when we zoom in, then you can see how there is this cleft in between the two cells. And this is where we're able to send a signal from the presynaptic cell to the postsynaptic cell. And the main way we do that are through these neurotransmitters. So these are molecules that are made in the presynaptic cell, and they're sent out into this cleft to then connect with receptors on the postsynaptic cell. And this will then activate the postsynaptic cell to either fire a signal or stop sending signals. Now, while there's neurotransmitters and these receptors, there's also some things that can block the receptors. So that's what different types of drugs do. So some drugs will stop neurotransmitters from bonding with the receptors. Some will stop the neurotransmitters from leaving the presynaptic cell. What cocaine does, if you're interested, is it blocks the neurotransmitters from going back into the presynaptic cell. And so that's kind of like it blocks the cleanup. Because once you send the signal, these neurotransmitters get pulled back into the presynaptic cell for recycling. So what are neurotransmitters? So we're going to talk about some specific neurotransmitters that you're going to want to know, as well as just in general what neurotransmitters do. So neurotransmitters are chemical substances that carry a message across the synapse to either excite or inhibit other neurons. So when you excite another neuron, what we're talking about is causing the other neuron to fire, so it will create action potentials. When we talk about inhibiting firing, then that's for cells that constantly fire. So what we're doing is we're telling the cell, okay, you can stop firing now. So we have 5 stages of the chemical communication. You're going to want to know all 5 of these stages. So the first stage is synthesis. So this is we have to make the neurotransmitters. And this is done usually in the cell body or the soma of the neuron. Sometimes it can happen at the terminals themselves, but it's usually done in those somas. Step 2 is storage. So once we have the neurotransmitters made, then we're going to hold them in what we call synaptic vessels until we're ready to use them. So they're kind of just packed into, kind of think of a suitcase. We just put them in a suitcase or a box until we need them. And then release is where we take those synaptic vessels and we push them outside the cell so that the neurotransmitters within can move into that synaptic space. Once the neurotransmitters are in the synaptic cleft, then they're able to bind. So binding is step 4, and this is where they attach themselves to specific receptor sites on the postsynaptic cell. And then step 5 is deactivation. And what this is it stops the neurotransmitter signal. And usually deactivation will be that kind of recycling I was talking about before. So there's some other chemicals where their job is to go into the synaptic cleft and collect the neurotransmitters and recycle them back into the neuron. So here's an image of these different steps. So we have the in the presynaptic neuron here, our nerve impulse is sending down to the postsynaptic cell, and our synthesis of neurotransmitters again is usually happens in the soma or in the cell body of the postsynaptic cell. And then the neurotransmitters will be sent down the axon to the vesicles within these synaptic vessels. So these vessels help store them, and they also help move the neurotransmitters to where they're needed in the axon terminal. When they're needed, then the vessel will fuse with the cell wall and release all of those neurotransmitters into the synaptic cleft. So each of these little red circles is an individual neurotransmitter. The neurotransmitters will then bind to specific receptor molecules on the postsynaptic cell. And so this would be the dendrite of the postsynaptic cell. Now, what this image up here on the right is showing is that different neurotransmitters will have different receptor molecules that they'll bind with. So you can kind of think of it as a lock and key situation. So you have to have the right key in order to open the right lock. Here we have this neurotransmitter with some triangular green binding areas, and the receptor molecule matches that. However, the neurotransmitter on the right has this square binding section, which does not match their receptor molecule, so it won't be able to bind. It's going to have to go find its specific receptor. And this will come into play when we start talking about all the different types of neurotransmitters. Now, once we have binding to the receptors, that'll kind of happen for a bit, and then what's going to happen is you're going to have other cells or other molecules will come in, either from the side, or they'll be released from the presynaptic cell to go in and collect all of these extra neurotransmitters and bring them back up into the presynaptic cell. And from there, then they'll either be broken down or just recycled back into synaptic vessels so that they can be used again. So I mentioned before that there are two different types of neurotransmitters. We have excitatory neurotransmitters and inhibitory neurotransmitters. So for inhibitory neurotransmitters, what this does is it decreases the resting potential of the presynaptic or postsynaptic neuron, so at hyperpolarization. So that means we're getting more negative. So we're going from negative 70 to about negative 90, okay? So that's what inhibitory does. It makes it more negative, and that means it's harder for the cell to fire, right? Because remember, we need to reach the threshold for an action potential. So by lowering the resting potential of the cell membrane, then we're inhibiting the cell from firing. The opposite of that is an excitatory neurotransmitter, which increases the potential of the postsynaptic neuron. So it's going from negative 70, so remember that's going that way. This one will increase to, I mean, it depends, but it could even increase to that negative 50, which remember, this is the threshold for an action potential. So by doing an excitatory neurotransmitter, we may even get the cell to fire because we're increasing the resting potential from negative 70 to negative 50. That's a negative. Okay? So I want you to keep that in mind. So again, here's a nice little diagram of what's happening. You've got an excitatory neurotransmitter. It depolarizes the neuron's membrane, which increases the likelihood of an action potential. The opposite is you have an inhibitory neurotransmitter. It hyperpolarizes the neuron's membrane, so it becomes more negative, which then decreases the likelihood of an action potential. So from this, we're going to move on to some specialized neurotransmitters that you're going to need to know specifically. So I'm going to end this here, and we'll have the specific neurotransmitters on their own lecture video.